All right, so what we're going to do next is look at the Hardy-Weinberg theorem. And Hardy-Weinberg is not one person, it is two, Hardy and Weinberg, and they both came up with this idea at the same time. So they're both given credit for it. So what you're looking at is ugly. Let me just say that. I'm not sure how helpful the pictures are. But I do want you to realize what we're kind of talking about when we use this theorem or theory. And so what they're trying to show you is over on the left is the frequency of the CR allele. So they're looking at the dominant allele versus a recessive. How many red circles do we have versus white circles? So if you do the math, the frequency of the large P, they say is 80%, and then or the red one is 80%, and then the white one is 20%. And so when you make egg and sperm, what are the chances of that getting passed along? And the one on the right is trying to show you in the crosses, what are the percentages and how can we do this to get an idea. So I'm going to show you a slide next, and I might as well show it to you now, that I need you to memorize. What we're going to do is we're going to look at ways of doing what's on the right here. I think simpler than what they're showing you. But I'm going to do it in class. But what I need you to do is I need you to kind of memorize these two equations and what they mean. And I'm going to tell you on our next test, I'm going to ask you what some of these stand for, just the letters, because it's important. And so the equations you have to memorize is P plus Q equals 1, and then P squared plus 2PQ plus Q squared is equal to 1. Now, the first one is obviously, hopefully those are simple to remember, is the P is always going to stand for the frequency of the dominant allele, and Q is going to be the recessive allele. So in the last picture, the P was standing for the circles that were red. That was obviously the dominant trait. And then Q was going to be the white one. And of course, that has to equal 100%. It just has to. Okay. The second equation is the binomial expansion of the first one. And you're going to notice there's a ratio there. 1P squared plus 2PQ plus 1Q squared equals 1. That is that 1 to 2 to 1 ratio that we see in a monohybrid cross for the genotypic ratio. And so P squared is the frequency of the homozygous dominant allele. So if we're using the letter R, two large R's, PQ is the frequency of the heterozygotes. So that would be large R, small r if we're going to use the same letter. And remember that we would have two boxes in the crosses for that. And then Q squared would be the frequency of the recessive, homozygous recessive individuals. And so I need you to memorize this. So we're going to go through and look at this and what this does just kind of in general. And then again, we'll work this problem out in the Zoom, probably on next Wednesday. Is that I want you to understand this is a way of trying to explain the percentage of the population that has a certain type of genotype. And so if I walk around and I look at the group of people, let's say I have 100 people just to make the math easy, and 25 of them have blue eyes. We mentioned that blue eyes would be that recessive trait. Then 75 would have brown eyes. So I would know what that population is. The problem is with everybody with brown eyes, I wouldn't know, the carriers, how many of them are how many little bees are out there in those brown eye population. And this is a way of calculating it. And we're going to show you that. Here's the problem with the Hardy-Weinberg theorem. It works great, but it is a snapshot. It is giving you a picture of what's happening at the moment. And it is also not actually telling you how many of those brown-eyed people are carriers of the blue-eye trait. I might end up knowing that 50 of them are carriers. I just may not know who they are. Now, if I look at parents and grandparents, I might be able to deduce some of those. But this is a way of just kind of getting, again, a snapshot of what's going on in the population at a particular time. With that being said, there are assumptions that this theory only works if there's a large population, if there's no gene flow, and gene flow is the idea of individuals entering or exiting the population. So if something's coming into a population, we call that immigration. If something's leaving, we call that emigration, EMI. I asked that once in class, and students said deportation. Hopefully that's not the case. Okay, no net mutations, which means that there is not a mutation from one to the other. No natural selection. So every individual has the same chance of survival, and random mating. And what that means is that there is no selection pressure being put on any phenotype that you see, that we're not picking for a particular brown eyes or blue eyes or anything like that, because if that is being done, basically evolution is occurring, and we can't get a good handle of what's going on. That's why I say this is a snapshot, because I will tell you that these assumptions never are all met. It's just not going to happen. But it can be a useful tool to get information at any given moment. So again, we're going to do this in the lab, and we're going to do this in the Zoom on Wednesday, I believe. So I think that will be helpful. But for now, I just want you to memorize those equations and think about the assumptions. And so with that, we'll stop the share and stop the recording.